Good afternoon, everyone. It's my pleasure to welcome you all to the Treesearch Research Infrastructure webinar series. My name is Anastasia Riasanova, and I'm a coordinator of the Treesearch Research Infrastructure. And today it's a great pleasure for me to welcome my colleagues from Chalmers, who will give you an overview of the infrastructure facilities that are available at Chalmers through the Treesearch Research Infrastructure. And I would like to start from Aleksandar Matic, who is a coordinator of the infrastructure node at Chalmers. So please, Aleksandar, the floor is yours.
So thank you very much, Anastasia. And it's a real pleasure for us to introduce infrastructure that all of you active in Treesearch can access and benefit from. And it's really pity that you are not here, because Gothenburg is at its best today. We have a wonderful sunny day, and you would really have enjoyed it also actually to come to the labs and see things. But welcome anytime if you have any questions or just want to see how the labs look. Please don't hesitate to go down to Göteborg and visit us. We put together a small program for you to introduce the infrastructure. So I will first make a little bit of an overall introduction of what we can offer at Chalmers and also what you can expect from us. Then we go into details in three of the techniques, the three main techniques that we offer. So small and wide angle X-ray scattering. So Polina Naidjonoka will introduce this for us. Raman Spectroscopy. There we have our expert Nataliia Mozhzhukhina. And then static and dynamic light scattering, then Polina returns to explain that part. These are also two of the persons you would most likely meet if you come here to do an experiment with us. There's also a few more that can that might assist you. And in the end, I will wrap up a little bit. And all the three of us are at the Department of Physics at Chalmers. So looking at the Treesearch infrastructure that can be accessed at Chalmers, it's actually divided in two locations. One is the Chalmers Materials Analysis Laboratory, and the second one is the equipment we have at the division where me, Nataliia, and Polina work, the division of materials physics. And if we go to if I start with the Chalmers Materials Analysis Laboratory, this is actually a central facility at Chalmers. So it's a facility open for all researchers. Where we have technicians who take care of the instruments. There is one booking portal for all instruments where you can see if they're available. Also, this facility provides training and students for postdocs, well, also senior researchers, if you like, to become direct users of the instruments. What all of you should be aware of that are at other universities, and for that matter, companies, is that this facility is open on equal conditions for all academic users, which means that if you come from KTH or Mittuniversitetet or Umeå, you will actually pay the same fee for the instruments as I do. So there is no differentiation. So it's really an open facility. Also industry have access, but of course you pay another fee in that case. You go to let's see if this works. Yes, it works. If you go to the web page of the Materials Analysis Laboratory, you can see here, you could go under Instruments, and there is information on the details. There is also in general contact persons. And through this laboratory, you can get access to more instruments than what we offer directly through Treesearch. So I really urge you to go to the web page of Chalmers Materials Analysis Laboratory. And you have the web page down here. So the instruments, we mainly offer the instruments where we provide some kind of support here at the Chalmers Materials Analysis Laboratory. It has to do with electron microscopy, so ion beam techniques, so FIBSEM, or environmental SEM, and also X-ray scattering. So we have several really nice and new instruments on the electron microscopy side. I understand that they are so nice, so even people from KTH are tempted to go to Chalmers. And as all of you know, to get somebody to leave Stockholm to go somewhere else, that takes a lot. So this is, I think, a good sign of the quality of our equipment. And one instrument which is especially interesting is this TESCAN instrument, focused ion beam instrument, which can really provide high resolution images also for soft materials, combining a low current and low voltage and really optimized detectors. You can also reduce these charging effects that you can get in soft matter through this gas injection system with carbon that is available. And one recent example we had here is work done in a project by Cecilia Fager, Per Larsson, and Lars Wågberg from KTH, where they wanted really to look on modified cellulose fibers and then using FIBSEM. And you can see here images really taken by Cecilia, very high quality images where she has managed to take super nice cross sections with this FIBSEM. And then the idea is to still look for the effects of modifications. And here they also use a staining technique. And here we should also point you to Cecilia, who is the big expert. She has now left KTH and Treesearch, but is still very interesting in this area. So if you have any questions about this or want advice, as I understand, Cecilia is really eager to help, and is also starting her own company actually in this area. So I have her contact details here. The other instrument which we are offering through the Chalmers Analysis Laboratory in the framework of Treesearch is, of course, small and wide-angle neutron X-ray scattering instrument, which also in principle has a GSAXS option, even though that one is not that much used. But this is really a nice instrument where you can do a lot of different example work related to Treesearch materials, and Polina will introduce us to this in a minute. I think what's interesting for you is what kind of support you can get here. So looking at the instruments that we offer most, so small and wide angle X-ray scattering, Raman, and static and dynamic light scattering, the help you get from us is advanced user support. So we have persons who will advise and help you in planning and setting up of the experiment. They can also assist you with instrument training, or in the case you have just a few samples, this can also be a direct measurement service. We have one experiment coming up now with RISE in Borås, where they only need Raman spectra of 3 samples. Doesn't make sense to have somebody trained. They just send us the samples, we return the data to them. But it can also include analysis and interpretation support. Sometimes if an experiment is particularly complicated or new, this can even contain development of analysis models. There is one example where we worked with Stora Enso on the new materials, and we developed a little bit new analysis approach to get the information that was needed. This service of the advanced user support is done for free, but what you have to do is you have to pay the user fees if you use the SAXS and WAXS instruments. If you use Raman or light scattering, then everything is for free, as long, of course, as your project is approved by Treesearch. For FIBSEM and ESEM, here we mainly accept expert users because we don't have the advanced user support on site. So as you saw, the project that I demonstrated just a minute ago with Cecilia, then Cecilia was really an expert user coming from KTH, but of course trained at Chalmers. So if you are an expert users, we can at least to some extent then support the user fees on the FibSem or eSem. And I think if you're interested in these instruments, a good way is to approach Anastasia, who is a big electron microscopy expert or electron microscopy expert, and she could probably then work out with you how to use them in the best sense and maybe even accompany you to this experiment. What do we require in return, apart from your lifelong gratitude, is that you publish your results. So here are some examples from the group of Tina Nyppele and Annette Larsson, their groups. And as you see, it's really related to materials from trees, because everything is published in carbohydrate polymer. And you see that it's also usually a collaboration. So you see on the paper with Tina, there's a previous advanced user support expert, Barbara Berke, is part of that paper, similar for this one. And the one with Annette Larsson down here, then Martin was providing the support. And I think this is also a nice way where we develop collaboration. So with this short introduction, I think it's good we can continue in the program and dive a little bit more into the details. And with this, I would like to give the word to Polina.
Yes, thank you so much, Aleksandar. I just want to give a big thank once again to you. Sorry for interrupting you, Polina. And I just want to remind all the participants, if you guys have any questions, please feel free to write in the Q&A or raise your hand, and let's keep this as a dialogue. And with this, let's welcome Polina. So Polina, would you like to try to share your slides with us?
Yes, just a minute.
And sure, as Aleksandar said, if you have any questions, please feel free either to approach all of us directly as a specialist or talk to me. And yes, well, most of you know me as electron microscopy specialist. So with that, I definitely would be able to help practically also. Yeah. So.
Okay, sorry, I have some issues with...
No worries, this is fine. So let's check if maybe someone has a question directly. Just, yes, raise a hand or post in the Q&A. Now it works. So, Polina, we can see your slides.
And you can see now it should work, right?
Excellent. Yes. So please, Polina, go ahead.
Okay, thank you. So hello, everyone. I'm Polina. And today I will tell you a little bit about both X-ray and light scattering that we can do here at Chalmers. So I will start with a little bit of background in X-ray scattering. So maybe a lot of you know what is X-ray scattering, but I will go slightly a little bit into the theory. So in a typical X-ray scattering experiment, there is a highly collimated monochromatic beam directed towards a sample, and most of it passes through and some of it gets scattered. So that is what we actually measure. Photons interact with electrons and provide information on the fluctuations of the electronic density in the matter. And basically, we get data on the intensity of the scattering as a function of either scattering angle, which is to theta, or a scattering vector, or q. And eventually, you get this 2D scattering pattern at the detector, from which you can either directly get some information, or you can transform it into a 1D scattering curve. And then depending on the measurement, depending on the Q range, we call it either small or wide angle scattering. So with SAXS and WAXS, you can measure structures from 0.1 up to 500 nanometers. Here with our in-house SAXS instrument, we can measure up to 200 nanometer structures, but of course there is always a possibility to go to synchrotron facilities, and there you can measure larger structures. So some of the properties that you can, or some of the information that you can obtain include cellulose crystal structure, which is done with wide angle X-ray scattering, and then particle shape and polydispersity. These are measurements in solution. From that, you can also obtain aggregation, find out whether you have attractive or repulsive interactions between particles in solution. For fibers, you can determine the thickness, structure, alignment in solution, or alignment in the flow, and so on. So as I already mentioned, with our instrument here, we can measure up to 200 nanometer size of the particle, either in solution, or we can also measure solid samples. So here are some of the sample stages that we have for our instrument. So for solid samples, you can basically just simply tape your sample to onto the sample stage and measure like that. We did that with our wood samples, like here on the right. We actually just taped them on the sample stage. In case you want to measure liquid samples or wet samples, then you can do it with capillaries, which come in a variety of different sizes. And you can actually measure even very small amount of sample, even just tens of microliters is fine. If you have gel-like samples or wet samples, it's possible to use these kind of sandwich cells, where sample is placed in between two windows here and then mounted on top of the sample stage. So we can actually measure a wide variety of different kinds of samples. And here is an example of wide-angle X-ray scattering. So here we actually measured wood samples that I showed in the previous slide. So those were spruce samples with different pretreatment methods. And what we wanted to see is the effect of the treatment on the cellulose crystallinity. So here is a very typical wide-angle X-ray scattering pattern, and this profile is very typical for cellulose. So there are different crystalline planes that are measured, and this is a spectra containing different crystalline planes from cellulose structure. And depending on what you're interested in, you can determine the cellulose crystallinity. You can also determine what is the amorphous part or amorphous contribution in your sample, if you have any. In cellulose samples, that's basically just non-ordered or amorphous cellulose. If you have a more complicated samples like wood, then you have contributions from hemicellulose and lignin as well. And these are you have peaks here very broad as compared to the crystalline peaks from cellulose. So we calculated cellulose crystallinity affected by different treatments on wood pieces. However, we did not really see the clear effect of the severity of the pretreatment on cellulose crystallinity, and that could be due to the fact that the crystallinity index is calculated both from the amorphous part as well as the crystalline peak. So basically, if you have changes in both crystalline planes and amorphous planes, then you would not see changes in only crystalline parts of cellulose. What else you can get from wide-angle X-ray scattering is a crystallite size. So this is the equation that one could use to calculate crystallite size. Basically, you need to know a constant, which for cellulose is usually 0.94, X-ray wavelength, then half height width of a diffraction peak. And then you can use either which one, and the center of the diffraction peak or the Bragg angle. So for the same samples, we determined crystallite size of cellulose, and then we saw a clear change in the crystallite size of cellulose with increasing severity of the pretreatment. And this was an indication, a clear indication of fibrillar aggregation in the sample. So then on to small angle X-ray scattering. So with wide angle X-ray scattering, we can see interatomic structure. With SAXS instead, we can see particles in solution or fibrils. We can see the arrangement, we can see the size of larger structures. And in general, the smaller the scattering angle, the higher or the larger structure you can see with the technique. So in order to analyze the data, so this is general scattering curve that one could obtain. There are several things that one could do to analyze the data. So mostly it's either model independent analysis or model fitting. So with the model independent analysis, one usually looks at the slope of the curve, which then can indicate on the shape of the structure. And that could be, if the slope is -1, then it's rod-like structure, -2, a plate, or -4, a sphere. There is also something called Guinier regime, from which you can determine radius of gyration or size of the structure. So these are all of the things that you can use to get some kind of indication on your structure. From that, you can then go on to model fitting, or if you're satisfied with this analysis, that is also perfectly fine. But if this information is not enough, one could always use models that are already can be found in different softwares, or one could also write their own model. There are very wide variety of models. You just need to know which one to use for your sample. So there are models for spheres, cylinders, they can be core shell or Gaussian coils, swarm-like chains, and so on, and fractals. So you just need to know some kind of information about your sample before, in order to make an informed decision of which model to use. So here again, the sample of the spruce samples from the same project with Fabio Caputo and Lisbeth Olsson. So we measured also small angle X-ray scattering for these samples, treated with different severities. And here is a typical scattering curve. One would think that there is not much to look at, but it actually gives quite a lot of information. And the most important for us was one particular peak at this Q range, which actually indicates distance between fibrils. And what we saw was, with increasing severity, this peak would shift to higher Qs, which also means that the distance is increasing between the fibrils. So we determined from this study that the fibrils are aggregating, which we saw from the crystallite size increase, and that also forces the fibrils to, or fibril aggregates actually, to be further apart from each other and create pores in the structure. Another study that was done with SAXS at Chalmers was by Beatrice Svensson and Barbara Berg here. So they studied cellulose dissolution in aqueous hydroxide-based solvents. And so here you can see the scattering curves of microcrystalline cellulose dissolved in different solvents. They used model fitting with semi-flexible polymer with excluded volume. And what this model gives is a contour length. It gives indication on the stiffness of the polymer chain given by Kuhn length and persistence length. And it also gives radius of the chain. So with this, they could see that in some of the solvents, the polymer chain is more stiff and more aggregated as compared to other solvents where it's this dissolution is more preferred and the polymer chain is more flexible. Thank you. Do you have any questions?
Thank you so much, Polina. We have one question from Shijil. I think I pronounced it correctly. If not, please apologize me for this. So, while calculating the crystallinity index, why the intensity of 200 plane ID preferentially selected? Maybe you can also see the question in the chat.
Actually, this is what is done mostly in literature, but one could use also other planes. I think this is, the peak is more intense, so it gives a more accurate result. But in many cases, people compare different contribution from different planes and then compare the crystallinity index. So it's completely possible to do it with other planes as well.
Hello.
Hello. Yes, I asked you to unmute, so please go ahead and join the discussion.
Okay, thanks for that. My name is Shijil. So I'm going to join in Luleå University as a PhD student. Okay, my question, I asked that particular question because my interest is that how you are measuring how the 200 plane intensity goes on decreasing or increasing, increasing or decreasing with the amorphous phase. What was the reason for that particular crystallinity index checking?
Well, the idea initially was to follow the changes in the crystalline plane and hopefully to see either increase or decrease. But actually, we did not see like just a trend that follows with the increase in the severity, because with these treatments, you affect both the cellulose fibrils as well as the amorphous parts of the sample. So we could not conclude which one it is, if either we are disrupting the cellulose change and increasing the amorphous part of the sample, or we are dissolving some amorphous parts, and then you would expect that crystalline parts or the percentage of crystalline parts would increase. So that was a little bit inconclusive.
Okay, so we have to choose the plane with maximum intensity for the calculation. That means here you have preferentially chosen 200 plane for the calculation.
Yes, that is something that is most oftenly done.
Okay, thank you.
Thank you so much. Are there any more questions to Polina? Not right now, maybe. So Polina is still here and she will present a bit later. So if you get any more questions, please feel free to post in the Q&A or in the chat. So,
yes, absolutely.
Yes, thanks again, Polina. And now it's time for us to move to Nataliia Mozhzhukhina. And Nataliia will talk about Raman Spectroscopy. Nataliia, did I pronounce your last name correctly?
I didn't hear it because the sound just went off. So.
Okay. So Nataliia, we welcome you to talk. Nataliia Mozhzhukhina, right?
Yes, that's correct.
Okay, correct. So good. Nataliia will present the Raman spectroscopy. So Nataliia, the floor is yours.
Thank you. Okay. Great. I hope you see it in full view. If not, just let me know. So thank you very much, Anastasia. Yes, my name is Nataliia Mozhzhukhina, and I'm a postdoctoral researcher with Aleksandar Matic here at Chalmers at the Department of Chemistry. And it's my pleasure to give a presentation about Raman spectroscopy and also what kind of use it can give for the wood science research. I will give a general presentation into the Raman spectroscopy as well as our availability here in Chalmers, and also give some concrete examples of its use by some measurements that I did, and also measurements from Åke Henrik Clemens from the Department of Chemistry. So the Raman spectroscopy, it's recently celebrated, actually, Raman effect, its anniversary in 28th of February, I remember, and it was invented in 1928. The effect was discovered by Sir Chandrasekhara Venkata Raman, which is a scientist after him. Technique is called like this. He was Indian physicist and was awarded in 1930 Nobel Prize for his discovery of Raman spectroscopy, and he was actually the first person in Asia to receive this kind of prize. So Raman spectroscopy is based, as many other spectroscopies, on the interaction of light with a matter, and in the case of Raman scattering, this is effect based on inelastically scattered light from a molecule. Sorry. Normally recorded in as a backscattered Raman, also could be a forward scattering Raman effect. So it detects an elastically scattered light from molecules. In the case of molecular vibrations in the molecules, we typically detect the bonds and can identify the bonds between molecules, while in the case of crystals, Raman is also very useful since it defines a collective phonon modes, collective lattice vibrations. Also, just think to have in mind that Raman is by itself is quite a weak signal. Only 1 in 10 million photons is actually elastically scattered, which also means that the technique is not surface sensitive. I pointed out because very often people ask about how far and deep into the sample we can get a Raman result from the sample, and I will get into more details of this here. So as I mentioned, Raman spectroscopy is not surface sensitive, non-destructive technique, very useful technique. It probes the depths from hundreds of nanometers up to microns. I'm sorry for this. And the penetration depth of the Raman depends of the laser inside of the material, depends on several factors, which is calculated by this equation. So it depends on laser wavelengths. The higher wavelengths, nanometers, more deep is a penetration to the sample, and it's irreversibly proportional to magnetic permeability and electronic conductivity. So this means less conductive samples would also have a deeper penetration of the signal inside, which is a case with actually wood materials rarely conductive enough. And Raman spectroscopy provides information on the chemical composition of the sample, of solvation structure, crystalline structure, short range order, degree of crystallinity. All these informations can be fished out of Raman spectroscopy if analyzed in a good way. At Chalmers, at the Department of Physics, we have 3 Raman spectrometers that are shown in the images here, one of which has its own advantages or disadvantages compared to the other one. We also have availability to do confocal Raman spectroscopy, which means that this is Raman that is connected to optical microscope. So with this, we focus on the sample and can get a very low lateral resolution, and therefore a Raman mapping. We have a big variety of laser wavelengths available, including from green, blue, red, near infrared nanometers lasers, which also could be used for different materials. For example, we would like to use a higher wavelengths in the case of materials that are prone for fluorescence in order to reduce the fluorescence effect. Or we could also change to another wavelength that will be more in resonant with the material to have a better signal. And now, after the introduction of the technique, I will just present a couple of examples. So first of all, I will present examples of the Raman measurements done on our Raman machines by Åke Henrik Clements, a PhD student from the Department of Chemistry here. And he did Raman microscopy, so it is a confocal Raman or Raman mapping, all the same synonyms for the same thing, of the pulp fibers. And he could analyze the spatial resolved composition of these pulp fibers, including analyzing the degree of delignification, also the chemical alteration of lignin during the different processes such as cooking and extraction, and also the process of diffusion of the plasticizer in the cell wall. For example, of the specially resolved composition of the pulp fibers with Raman microscopy or confocal Raman, he used a lab RAM instrument with red laser and 100 objectives that does provide round micron, slightly below micrometer kind resolution depending on the hole used, and could qualify the semi-quantitative degree of delignification ratio of cellulose and lignin bond. So here you could see the spectra of the Norway spruce and mildwood lignin, as well as two different kinds of softwood kraft pulp. There are some very specific Raman signatures from both lignin and cellulose, and this could be resolved at different parts of the sample of Norway spruce, or on the inner, or in this different walls of softwood kraft pulp. And as mentioned before, with this it is able to quantify lignin antillations during the processes. And there's, of course, in some of the samples, a lot of photoluminescence visible due to condensation and degradation of side groups, which could be again worked out with modifying the laser and intensity, and also a good background subtraction. Another example provided by Åke is the diffusion of the plasticizer in the cell wall. So this is also a scan, and the same scan of Raman measurement with the baseline subtracted, where also one can differentiate the peaks and signature of PHG, cellulose, and lignin in the peak treated kraft fiber. And pig is appearing here. Now moving a little bit from wood to carbons. So the lignin and cellulose materials, if they're heated up in a correct atmosphere to correct temperature, provide very nice carbon materials that are called hard carbons, that are interesting materials for energy storage applications. And the carbons is also particularly actually good to be studied by Raman Spectroscopy. This is one of the example of hard carbons that are derived by this thermal treatment of lignin materials that are characterized by very visible two peaks, but actually the good fitting of these carboxygenous materials are represented by 5 different peaks, 4 D bands and 1 G band. And each of them corresponds to a graphitic domain and also amorphous carbon domains and impurities. So in this way, we can characterize these hard carbon materials. We can get the ratio between ordered and disordered graphitic lattice, as well as presence of some amorphous sp3 or graphitic sp2 domains. In plain graphite crystallite size could be determined, which is determined actually by this formula, conic equation, as in plain crystalline size, graphitic depends on laser wavelengths and the intensity ratio between D and G band. In this case, D is D1. And also we can get the idea of the some presence of surface groups, CH termination, adsorbed molecules, etc. in the material. To go even one step further than this, we not only have Raman measurement and mapping, but a variety of specially designed cells to do Raman in situ while varying parameters, as for example, temperature or also potential, which are in situ electrochemical cells. So these are examples of in situ electrochemical Raman measurements when hard carbon is used as example of the lithium-ion battery anode. And in this case, we cycle it versus lithium, looking at how these lithium ions get into and outside of these hard carbon materials by different processes that includes both insertion between graphitic planes, but also the adsorption in the pores. And this is an example of this spectra, how it looks like. In this contour graph, where here you have wave number and here's the time, and we see how the signatures of GNG band evolve within the time simultaneously to electrochemistry that is performed and controlled by potentiostat. Here we see the potential versus time as well, and how accordingly spectra change with lithium insertion and the insertion. And then we can perform the 5 peaks fitting and see the evolution of the bonds in the lithiated and lithiated sample, and as well as this bond positions and intensity modify within time, just as another example of the facilities that we could do. Also, it is not easy to fit many Raman spectra, like one can fit a couple, but in case of this kind of in situ measurements, when we take many measurements as a function of variable, would it be time or temperature, potential, whatever. For this, we have in collaboration with the partners in DTU developed and published a software that is available for free. It's a quick fitting tool called Prisma. It is available here at github.com bigmap Prisma. Bigmap because it is a part of a big European project, Bigmap. And we have published also a paper that describes how this software works, and it's published in Chemistry Methods. It is very useful for fitting. It performs Gaussian, Lorentzian, and pseudo-void fitting in semi-automated way on a big amount of spectra, and as a result gives parameters of the peak as a function of variable. And with this, I would like to end my presentation. Thank you very much for your attention, and I will be happy to support with the measurements and answer any questions if there are any.
Thank you so much, Nataliia. Really nice presentation. Let's see if someone has a question, please post it in the chat or Q&A or raise your hand. Let me check that I didn't miss anything. I don't see any questions yet, but I guess people will probably come up with the questions a little bit later. So maybe we thank Nataliia once again, and we will move on to Polina. And Polina will talk now about the dynamic and static light scattering.
Hello again.
I also want to encourage all of you, if you have any questions later on, you can always send an email to all the presenters and also to all the coordinators. Hear me well? Yes, yes, Polina, we can hear you very well. Yes. Yeah. Okay. Good. So please, Polina, the floor is yours.
So now I will continue talking about light scattering instead. So this instrument we actually have here in our department. So it looks like this. It looks a little bit scary, but it's actually not that scary. So what we have here is the light source, and it's actually very similar to X-ray scattering, where you have a light source that's actually hitting the sample. This is the sample chamber. So you insert a capillary with your sample. So this technique is only for liquid samples. So in this chamber, you insert your capillary with the sample, and here there are detectors that detect the scattering at different angles. And there are multiple varieties of these kind of instruments. So here in this instrument, we have different channels with detectors that detect different angles. It's also possible to have an instrument with a goniometer where detector is actually moving across to catch scattering from different angles. It's also possible to have instruments that measure only at one angle, like zetasizer, I think is one of the most common ones. And there you measure scattering at, I think, either 90 degrees or 170 degrees. In our instrument here, we can actually measure angles from 30, I think, up to 170 degrees, all at the same time. However, in this case, you would need a little bit larger amount of a sample, so at least 1 milliliter of your solution, and then depending on the concentration of your sample. So what is dynamic and static light scattering, or what kind of information you can get from these techniques? Is from static light scattering is where you basically measure intensity of the scatterings, very similar to small angle X-ray scattering. And from that, you get radius aggregation of your particles, as well as molecular weight, also possible to obtain. In dynamic light scattering, you get a correlation of the intensity, and from that you can then determine hydrodynamic radius or polydispersity of your sample. So a little bit into details about the techniques. So with dynamic light scattering, basically you measure the intensity of the particle, and it's actually based on a Brownian motion of the particles in the sample. And what you get is basically what you see is the movement of the particle in solution. So the larger the particles, the slower their movement, the smaller the particles, the faster their movement. And then from this curve here, you get a correlation between different points in time to see whether the particle has moved or not. And this is seen in a correlation function, which is usually calculated by the instrument itself. And then if you have small particles in solution, you get a correlation function with fast decay, or if there are large particles, then it's a slow decay like here. So from the correlation function, usually one would fit the correlation function with a cumulant fit, from which then you get translational diffusion coefficient. And then from that, you can get a hydrodynamic radius or diameter. And this is called the Stokes-Einstein equation. It's very familiar with the dynamic light scattering. So in order to use this equation, you would need to know the temperature and viscosity of your solution. Then static light scattering, instead, you just measure the intensity of the scattering, similar with SAXS. So large particles scatter much more intense as compared to small particles. And there are multiple ways how to treat the data. The most common ones are Guinier analysis and ZIM analysis. So in the Guinier analysis, you can simply just use this equation and plot this in a plot, and then determine from the slope the radius aggregation of your particles. You can do similarly with ZIM plot. However, the advantage of ZIM analysis is that if you measure at different concentrations of your sample and different angles, from this kind of plot, you can also determine molecular weight or apparent molecular weight of your particles. In addition to size, you can also get some kind of indication on the shape of your particles in solution by combining static and dynamic light scattering. So this is called shape factor, which is basically ratio between radius of gyration and hydrodynamic radius. Based on the value, you can determine whether it's a spherical particle, it could be a self-avoiding coil, or it could be rod-like conformation, and there are even more values like that in literature. So this is one of the examples of the results that you could obtain with light scattering. So in this study, we measured galactoglucomannan, which is softwood hemicellulose, and we compared it to well-known galactomannans like locus bingam and guar gum. So we measured both dynamic light scattering and static light scattering in order to find out what is the size of particles in solution and also get an indication of the shape. So what we found out was the galactoglucomannans have an elongated shape in solution as compared to galactomannans, and this could be explained by the fact that galactoglucomannans actually have side groups which make the backbone much stiffer as compared to galactomannans. Another study that was done here, again from Beatrice Svensson, on the cellulose dissolved in different solvents that I already mentioned before. So here they also did static light scattering, and they determined radius of gyration of microcrystalline cellulose in different solvents, as well as molecular weight from the ZIM plot. And they could see that in some solvents, radius of gyration is much smaller as compared to other solvents, and similar with molecular weight, indicating that molecularly microcrystalline cellulose would better dissolved in those solvents. Another parameter that you can determine from ZIM plot is A2, or second virial coefficient, which gives an indication on the interactions between the particle and solvent. If this value is negative, it means that there are repulsive interactions, and if it's positive, it means that there are attractive interactions, which they found that there are negative repulsive interactions in some solvents, but much more attractive in others. So these are one of the many things that you can actually determine with light scattering, which is very useful and can be very good technique to use together with, for example, small angle X-ray scattering to give an indication forward, for example, to what kind of model you would need to use to fit your data. And that's it about light scattering. Anyone has any questions?
Thank you so much, Polina. Really nice presentation. So, guys, don't be shy. If you have any questions, unmute yourself or raise a hand or post in the chat or Q&A. I don't see the questions right now. Good. Maybe I can ask a question, for instance, for the DLS and SLS studies. So our cellulose nanofibrils or cellulose nanocrystals, they usually have the filamentous shape, right? And to the best of my knowledge, DLS and SLS is working quite well for the spherical particles. So what is your reflection in terms of how reliable, how well it works actually for the CNF and CNC for the filamentous particles, how much we can trust the values and how much we can correlate it to the, for instance, the data that we will get from microscopy also.
So actually, most commonly used equations are all by assumption that you have a spherical particle. But it is possible to use other equations that are for cylindrical, for example, particles. And then you can also go more into detail of the analysis of your data if you know that you have something elongated. So actually, I think static light scattering works very good with elongated particles. You just need to know what equations to use and what analysis to do, but it's definitely possible.
That actually proves that you don't need to do it alone. You must be working with an expert who knows how to do data interpretation. Yes, we have a question from Edwin. Let's see, maybe I can allow Edwin to talk. So, yes.
Well, can
Yeah.
you hear me? Yeah.
Yes,
Perfect.
so Edwin, please go ahead.
Well, thank you, Polina. Thank you for your talk. I was just curious because you said that one can use light scattering to determine the molecular weight of cellulose in solution. And I was just curious to know if it's also possible to learn the molecular weight distribution. So like get a grip on how many of a certain molecular weight there are in solution and sort of like the distribution, if that's possible or not.
I think that is not possible because you also get an apparent molecular weight.
Yeah.
So I think from the analysis, you only get average molecular weight.
Yeah, sure. Do you know what kind of average we're talking about? If it's weight average, number average, or some other average?
I think it's number average, but I'm not
Okay.
completely sure at the moment.
But thank you. Thank you.
Thank you so much, Edwin, for the question also. Any more questions? No? I actually would like to ask a question to Nataliia now about the Raman field. So I have quite many colleagues at KTH who are working with transparent wood. So basically it's delignified wood which is impregnated with another polymer. And I have heard that using the Raman techniques, Raman spectroscopy is a very, very powerful technique to, for instance, see distribution of the polymer in the cell wall and in the lumen. Can you please comment on it? Maybe you have some experience with studying such kind of samples also.
Yes, so no, unfortunately, my experience is only widely to this carbonaceous material or bitazar, so it did not work directly with this kind of samples. But definitely what I can say, you are able to differentiate this kind of polymers and they have specific signatures, and they do. And then what you need is a good Raman mapping with a good resolution, so sub-micrometer. So if depending on the size of this features, for sure in micrometer range, you can have a very nice mapping and see where is what. So yes, definitely.
Let me
just tune in a bit to avoid fluorescence.
Let me ask you about the requirements for the specimen. I guess the surface of the specimen must be as flat as possible, right?
No, not necessary, because no, you can also no.
So some roughness is allowed. That's good.
Yes,
because
yes.
We have some, you know, like it's a little bit tricky to cut the wood-based materials and to get the ultimately flat surface. So that's a big benefit. Yes, I get these questions quite often from my colleagues, how to clearly visualize the presence of the polymer in the cell wall and in the lumen. So basically now what I will do, I will direct them to the Raman specialists. They also do this research a little bit at other nodes, but I think it's good to cooperate also with you on these topics. That's great. Nice. So Aleksandar, what do you think? Should we move on?
We can move on, and we have covered basically our program. So
If maybe I can add a little bit also in terms of what was presented about the infrastructure, research infrastructure at Chalmers. There were questions about how to get access and training, and I would like to follow up this discussion. Like you said, Aleksandar, we are organizing the courses and trying to organize even more courses to provide the training and access to the people. So for instance, we started now with electron microscopy courses, the AFM courses on the way, then we will also think about some other fields and topics. And also, for instance, as we said before, you can talk directly to the specialists and discuss what would be the best. So usually the easiest, if you have just, a need for the proof of concept test, you will get the help from the specialist. If you find this technique applicable to a wide extent for your research topic, then of course you will be able to get the access to the facilities to a more large extent. So you can get the training and you can become like a regular user. And for instance, what comes to the electron microscopy, you can also always talk to the group of Eva Olsson and Stefan Gustafsson. I can see also Maria is here listening, Maria Brolo. So use your network and discuss with the specialists. Yes. So please, Aleksandar, go ahead.
Yeah, so that was very good. And I mean, also for the training, it's usually easiest when you actually have a problem, you have a sample you want to investigate, because then you will work with your own data, real data, and that's very useful. And as Anastasia says, just contact us and we can always discuss. So just to round off, remember that you have this possibility to get this support through Treesearch. You make an application through the web portal, and we are always pragmatic, so don't worry if you don't know too much about the technique. Submit your application, and the first thing we do is to start to discuss with you how the experiment can be arranged and if the technique is actually useful. Also, Anastasia has good overview over all the different nodes. So if she sees that your suggestion probably fits somewhere else, then she will direct you to that one. But I would also like you to, in connection to the small angle and wide angle X-ray scattering that Polina presented, just to highlight that through Treesearch, we also have access to the MAX4 facility through the MAX4 portal. So this comes through the fact that we have funded and will operate a beamline called 4MAX, where we also will provide this kind of user support. And one way to learn to use X-ray techniques at synchrotron facilities is actually to do in-house experiments, because once you get the data, it's not very different on how you analyze them. So the 4MAX portal is an access point to MAX4. It is centered around one beamline, the 4MAX beamline, which is a small and wide angle X-ray scattering beamline, but we will also provide access to other beamlines through the Treesearch portal. It's currently being built up, and the general user program is expected to start roughly in 1 year. So this is why I think it's very good to now start to do some lab scale experiments in order to be ready for this. And once ready, the 4MAX instrument will be very interesting in the sense that it will combine small and wide angle X-ray scattering. So as Polina showed you, covering, let's say, from the molecular scale to sub-micron scale. But then coupled to this, we will also have an imaging station, so to do X-ray imaging, X-ray tomography, where we overlap. So we will start from the micron scale and go up to the millimeter scales with the direct X-ray imaging. So think about this also as a possibility for the future, and maybe as a motivation to get further training in these techniques. So with this, I'm really thankful to my colleagues who have been giving you the scientific part, and also this opportunity for us to present what we're doing.
Thank you so much, Aleksandar. Yes. Are there any more questions? No? I think it's really good that you, Aleksandar, also presented the overview of the formats, especially closer to the end of this year. It will be up and running. So basically, don't underestimate this power and try to apply for the support. And exactly like you're saying, Aleksandar, doing the pre-studies on the lab scale for the SAXS WAXS, either Chalmers or also, for instance, using our neighboring research facility at Rice also. So yes, it will help significantly before you would do your real measurements at the synchrotron facilities, not only at Formax, but everywhere in the world. So that's why we need the lab scale SAXS WAXS measurements also. Good. I think it was really a fruitful discussion and really good overview, at least for me. I got a great update, and it was also nice to see you, at least remotely. We are planning to organize more such webinars. So all of you who are listening to us or who will watch this webinar later on demand, please keep your eyes open. We will post on the Treesearch website the schedule for the upcoming webinars. So basically the infrastructures from the other facilities will be also presented to you. And if you missed any of the previous webinars, please go to our YouTube channel and you can see these webinars recorded along with this webinar also.